Hello and welcome to this bioconference live presentation. My name is Shawn Baker and I'm the CSO of BlueSEQ. And today what I'll be discussing is how next-gen sequencing is driving the transition from genetics to genomics. So this is a fairly complex topic, and so I'm kind of expecting and hoping that you have some questions for me. And if you do, you can use the green Q&A button. If you click on that, you can then type in your questions. And at the end of the presentation, I'll try to take some time to answer as many of them as I can. So before we get started, I think it's important to say who BlueSEQ is and why you should be listening to us and what we have to say. BlueSEQ is an online sequencing exchange. We basically match together researchers who want to do sequencing with the providers who have excess capacity. But we actually don't offer any of the sequencing ourselves. We don't own any sequencers. We don't do any analysis. We're just a neutral online exchange. So we match people together. But since we don't own any of the technology, we feel we have an unusual, unique perspective on the market. We pay attention to what's happening today. We need to pay attention to what's happening in the future. But since we don't own any of the technology, we don't have a stake in the game, we can come at it from a very neutral standpoint. And that's that neutral standpoint that we're trying to share with you today. So before we get into how next-gen sequencing is actually transforming how diagnostics is being used and how genetics is changing into genomics, I want to take a step back and talk about where this market has been, where it is now, and where we think it's going in the future. So first we'll start out with who was driving this market. It was really the pioneers, the early adapters, the risk takers. So the people who were intensely interested in what the technology is, exactly how it works. They like getting the technology as soon as it's available. They like tearing it apart, seeing how they could make it better, even sometimes seeing how they could break it down. And they have a particular mentality, a certain characteristic set, and a certain types of things they're looking for in a technology and looking for in a market. And primarily what they wanted was dramatically increased output. That's what next-gen sequencing was offering. So this graph here is just giving a relative overview. It's not a complete history. It's just taking a look at one particular company, Illumina, and their instruments over the past several years. They started out with a machine called the Solexa 1G, and as the name would imply, it could output about 1 gigabase or 1 billion bases of sequence. But that rapidly changed once Illumina acquired them. It got renamed to the GA, or the Genome Analyzer, and the output was increased to 5 gigabases. Then over the years, it kept improving. The new model, the GA2, could go up to 30 gigs. That then got improved to the 2X, which was 50 gigs. Further improvements, primarily in terms of chemistry, but not so much the hardware, that actually got bumped up to 95 gigs. At the same time, they also released an even bigger instrument, the HiSeq 2000, which at launch could do 200 gigabases of sequence per run. That was then in 2011, got its latest upgrade, which bumped up the output to 600 gigabases. And internal runs were even bigger. They announced that internally they could get up to a terabase or so, or a trillion bases. And that's all in a single run. So in a span of about 4 years, we're looking at nearly a 1,000-fold increase in the output from these instruments. And obviously these early adopters just ate that up. Because you're getting these increased outputs with more or less the same cost per run, the costs are going to drop. So if you look at 2001, the cost per million bases sequenced was around $5,000 or so, and that was using pre-next-gen sequencing or Sanger sequencing technologies. And over the years, that slowly started dropping. That you can see that really precipitous drop there in the middle. That's when the next-gen sequencing market really started heating up. A lot of competition just really drove the prices down. So in the beginning of 2012, that same million bases that used to cost $5,000 then cost under 10 cents per million bases. So just a dramatic drop across this time period of 11 years or so. It's nearly a 5-order magnitude drop. So just tremendous tremendously less expensive to run the same kind of experiments. And of course, when you drop things so radically in terms of price, it really opens up things that you can do. So projects that weren't feasible before, all of a sudden people can start thinking about actually doing them, getting them done, trying new things they never would have even thought of trying before. So it was really empowering for these early adopters to just completely rewrite the way they were doing their research. So this, of course, resulted in very expensive machines that produced a whole lot of data and produce that data over a fairly long period of time, generally in the order of two weeks to a month even. So this was really good for big projects, researchers who had a lot of data that they wanted to generate. And the market players here, 454 was the initial next-gen sequencing player. Illumina came on the market a little bit later, followed very shortly by Life Technologies with their solid machine. But at this point, for the high-end, massive amounts of data per run, it was really Illumina that came out as the de facto leader. The other players are still in the market. You can still get 454. You see research being done on them. The solid as well is still out there, but they aren't nearly as active as the Illumina platform. So at least in this segment of the market, the race is more or less over until a new competitor comes on the scene. So that was sort of who kicked off this next-gen sequencing market in terms of the buyers. But the question is, now that those early adopters have adopted the technology, have started publishing papers, what's happening now? What's happening now then is more the mass market, the everyday researcher, the people who certainly are interested in sequencing, they want to use it, but they wouldn't consider themselves sequencing people. They are more interested in their own research. They just want to take what they've been doing, use the new technologies to make it better, make it faster, make it cheaper. And as I said, because this incredible increase in capability and decrease in price, it's opened up all new possibilities of what's available on the research side. So these people can then expand what they were doing. But again, they're focusing more on their research, getting their work done, simply using these next-gen sequencers as tools rather than really trying to dig into the details of the sequencer and what it does. So these people have different needs than the early adopter. So these initial instruments are extremely expensive. So anywhere from $500,000 up to $800,000. And then you even have additional costs for other ancillary instruments, data storage, things like that. So extremely expensive. That's fine for someone who's really dedicating their research to using these machines 24/7. But for the more typical researcher who wants to just advance his own program, that's too much. And so the market has responded with much smaller instruments. These are desktop instruments rather than things that can be so heavy they actually require reinforced flooring. And they're also much cheaper. So $50,000 at the low end to $125,000 or up to maybe even $200,000. But the point is substantially less expensive than what the market was serving before. The other thing that this segment of the market wanted were quicker runtimes. So, rather than having an experiment take 2 weeks, 3 weeks, maybe even 4 weeks, they want it to be done in hours, or at least within a day or two. And that's what these new instruments are providing. The other thing, of course, is looking for a lower cost per run. At the high end, these machines, a single run can cost as much as $20,000 or more just in reagents. And now with the new machines, they can be as little as a few $100s. So this makes it much more conducive to doing quick, rapid, low-cost experiments, doing a little playing around, seeing what you get, going back, looking at the data, and then revising and running another experiment. When you have a big, long, 2-week, 3-week experiment that costs $20,000, you have to be much more careful with the planning, much more certain of what you're putting in and what you're trying to get out. The other thing that really everyone's been looking for, not just the mass market, but also the early adopters, everyone wanted increased read lengths. So when the Illumina system came out, it was producing really just 25 bases per per read. So read lengths of just 25 bases. And this would be in contrast to around 1,000 bases or so, at least several hundred bases for the initial Sanger sequencing technology that it was trying to displace. So 25 bases obviously isn't nearly as long, isn't nearly as useful, but because there were so many of the reads, um, it still, people were still able to get a lot of good use out of it. But in order to increase that utility, they really wanted to expand on this and get these longer and longer reads. So it started out at 25 bases, was relatively shortly doubled to 50. Lately, we've seen 100 base reads and 200 base reads become common, and we even have had 400 base reads released on certain technologies. On some specialty instrument from Pacific Biosciences or PacBio, they even have average reads that are about 5,000 bases long, with a smaller number of those reads reaching into the 20 kb range. So clearly, once you start getting these longer bases, longer reads, again, it really starts opening up various applications that become possible. And so people are pretty excited about this. And there's been a lot of progress made, but we would expect even more effort to be placed here in the future. So the primary market players for this mass market segment are the ones we've talked about before: Life Technologies with their PGM and proton machines, and then Illumina with their MySeq machine. There are a couple other smaller players, but these are really the two key ones. And unlike on the high end, where Illumina really has won that race and dominated the market, um, this race is by no means dominated by either one. It's a highly competitive situation. Uh, both these machines are actively being improved. So it's actually a really good thing for the buyers out there, because these companies can't rest on their laurels. They really need to keep improving to make sure that they remain competitive. So the unfortunate thing, though, with what's been focused on over the past couple of years is that it really hasn't had much of an impact on sequencing costs. So while during the first 11 years or so of this graph, we showed a nearly 5 order magnitude drop in cost from 2012, the beginning of 2012 to about now, so about a year and a half, the pricing has really been quite flat. It even occasionally ticks up whenever one of the vendors increases their pricing, but we haven't been seeing that rapid drop in price that has almost been the definition of what next-gen sequencing is. So some people are starting to get a little bit concerned about this. It's probably not that big of a deal. It's really just a matter of these companies responding to what the market really wants. And so what this means is that the cost right now isn't the primary concern. But at some point, of course, it's going to become that concern. I expect to have increased competition in the future that will probably also help to drive this cost down. But for now, um, you really shouldn't be relying on those precipitous drops that we've been seeing over the past few years, because at the moment it's fairly flat pricing. So we talked about sort of who started this market and who's dominating now in terms of buying these machines and what they're using them for. But very soon, the question becomes, well, who's going to be driving the market in the future? And the future is more or less right now, and that's physicians and clinicians. So unlike the early adopters, they have absolutely no interest in the details of how the sequencing is working. They just want to see the results, even more so than what I would call the mass research market. They really just care about those end results, how the sequencing data is going to impact how they help their patients, guiding their diagnoses, and that's what they're focused on. So the qualities that they're interested in, they really want higher quality data. Everyone wants high quality data, but it's tantamount for the physicians and clinicians. And we can see from this graph, this is just showing a typical quality curve. So on the left-hand side, we actually see the quality scores, and then along the bottom, we're plotting the length of the read. And as the reads get longer and longer, the quality starts dropping off. And that's actually what the ultimate read limit is on most of the technologies. They could read longer, but the data becomes so poor quality that it just doesn't really add anything to the experiment, so they just kind of cut it off. And that mostly works fine for the research market, but for the clinical market, where they're really trying to look at potentially rare phenotypes or rare SNPs. They want to have a really low false positive and false negative rate. The high quality is extremely important to them, and that's what the market will likely be focusing on delivering over the next few years. Another thing is, I'd say, better and easier data analysis and interpretation. So what we're looking at here is a very typical routine file called the FASTQ. This shows the actual sequence data along with some information about each read, including the quality. So this particular snippet that I'm showing you is looking at just 3 reads from a run, but these runs can be anywhere from a few million reads up to 3 billion or so reads. So this is a huge amount of data, and this particular format, while incredibly useful for someone who um, can handle large data files, isn't very useful for an individual to start poking around and seeing what's going on with a patient. A more useful file would be the VCF, um, and this is basically a file that's looking at the variants. So rather than reporting all of the raw sequence, we're just looking at what variants are there. And specifically, we're saying what changes are present in this particular sample as compared to some common reference sample. And these are the places within the genome that the physician might want to look into, especially if it falls in line with a particular gene that might be related to the phenotype that patient has. So this is getting more useful, but as you can see, it's still difficult to actually glean anything by just looking at this raw file. They need better and better tools to get them where they really need to go, that is making the diagnosis. So data visualization tools are getting better too, and they'll continue to demand even better ones. And as we can see, this starts becoming a little more um, easy to interpret what's going on. At the top end there, we have an image of one chromosome, and in the middle, what we're looking at here is actually RNA-seq data. And on the bottom is a zoomed in view of one particular gene that we're looking at. And because it's RNA data, we can see that all of the reads are actually aligning to the coding regions of the exons, and very few reads in the intervening or intronic regions. The point is that this gives a much easier way of looking at what's going on. It's easy to see that these genes are here. I can see where the reads are, but we're only looking at one gene at a time. And while that can be useful if you know that you're interested in that particular gene, if you're trying to look at what's going on in all 20,000, this isn't particularly useful, or at least not useful enough. So where I think we're going to see the most improvement are the actual medical reports that come out at the very end. So here we're not interested at all what all that raw data looks like, even what all the variants look like. If you take a typical individual and compare them, sequence them, and then compare that data to the common reference sample, you're still going to get, several million variants, several million differences between them. And obviously, most of those aren't going to have any medical relevance whatsoever, but a small number will. And those are the ones that physicians want to see. They want to see that data in a very easy readable format. They want to see it in a way that lets them take that information and immediately apply it directly to their patient to say, okay, I know they have this particular phenotype. I see some lesions in these genes or these pathways which are related to that phenotype. This gives me a clue as to what the next steps are. Help me make that diagnosis. And so we're just starting to see that. A little bit later in the presentation, I'll actually show you some of the companies are actually at the forefront of this, making this information better in terms of what's presented and also in terms of how it's presented. Okay, so the market players that we would expect to see to serve this market, the physician market, is probably going to be the same. Life Technologies and Illumina are certainly both working very hard to try to make their products better for this particular market. But we're also likely to see many, many others come along. I mean, as you move away from the research market and move into the medical or clinical market, it just gets so much bigger, and that starts just attracting many new players offering services that just aren't offered by the current players. There's a lot of future technologies that are out there, people are working on, that might be the next things that maybe either displace life or Illumina, or even potentially get acquired by life or Illumina. One segment of that has been the nanopore companies. There's been a lot of news and a lot of excitement about these companies. They still haven't actually delivered on this yet, so this is still a little bit more experimental. But the idea is to take a nanopore, so just basically a small pore, either based on a protein like alpha hemolysine, or sometimes people make solid state ones. But the idea is that as the DNA molecule is drawn through that nanopore, it creates a change in the current, and that change can be associated, at least that's the hypothesis, is that change in current can be associated with individual bases. So if you could get this to work, you could have a very cheap, very fast, and potentially high quality sequence being generated. Um, and because of this great potential, there's a lot of companies working on this. And I'm just showing you some of the main ones here. But there's other strategies. There are a number of companies who aren't using nanopores, but instead looking at electronic detection. So as each base is incorporated or as each base is queried, it can have a different electrical signal. And there's a number of companies working on better ways of measuring those signals and turning it into high quality data. There are even companies that are looking at basically transmission electron micrograph or TEM-based technologies, where they're literally looking at each base individually, trying to make the call. The advantage of these is if they do it well, they could actually get extremely long reads. So I talked about typical next-gen sequencing reads being in the 150 up to 400 base range. With a specialty instrument from PacBio, you can get pretty big at 5 kb or so. But for these instruments, they're talking about starting at 50 kb to 100 kb. So really, really long distance reads. And that starts opening up new possibilities in terms of what applications are available, getting long range information, looking at rearrangements, things that right now are possible with other next gen technologies, but much more difficult to do. And then there's a whole slew of other technologies that are out there that are filling in little niches. But the point is that, as you can see from the slide, which isn't even complete, there are a lot of people right out on the horizon looking to make that next-gen sequencing system. So we kind of talked about the technology and how it's evolved and who it's been serving. And now the question becomes, how is this technology actually being used in the clinic? So before we do that, I kind of want to lay out this graph that I'm going to show, this image that I'm going to show repeatedly, and that's getting at the idea of what genetics versus genomics is. So on the left-hand side, we just have sort of a representation of all the chromosomes. On the upper right, we're zooming in and we're looking at just one gene. And in that gene, we have the coding regions or the exons, which are in blue, and then the green regions, which are the introns or non-coding regions. And then zooming in further on the lower right, we're actually looking at the individual bases. So just so you know that's what this graph is showing, we'll show it multiple times here. So kind of the quintessential genetic test is typically based on PCR, polymerase chain reaction. And in these tests, they look at a single locus. So that's what that red line is meant to represent on that one particular chromosome. We zoom in and look at the gene. Again, we're landing in one particular exon. And then zooming in once again, we're seeing that PCR diagnostic or test is looking at a particular base. So just one base out of the entire genome. Now, PCR can be multiplex, so you could actually get a few of these going together, but each individual test is actually only looking either at one base or potentially the presence or absence of a longer sequence. But the amount of data that's returned is pretty minimal. And as you can see in the lower part of the slide there, one locus with extremely low coverage, as would be expected. PCR really is primarily used in the diagnostic space for pathogen detection, like for HIV or hepatitis. It can be used to determine whether the virus is present. And also, if it is present, it can be used to determine viral load, which can be important for certain diseases. But it can be used for more traditional, what we can traditional human genetic testing. So one example of that is the EGFR testing. In this particular case, PCR is used to determine what particular allele of EGFR is present, and each allele will actually have a different prognosis for non-small cell lung cancers. So again, this is sort of a classic genetic test. We've got lung cancer. It's important to know, which version of that gene do I have, and depending on what you have, that's going to obviously change your prognosis, but also probably help guide your physician as to what treatments you should be receiving and how aggressive they should be. The other major genetic test would be the classic Sanger sequencing. So again, we're looking at one particular gene, as noted there on the left-hand side, looking at one red line going through the chromosome. But in this case, rather than looking at a single base, we're actually looking at what's probably the entire gene, or at least a very large chunk of it. Generally, Sanger is able to produce about 1,000 bases per run. So that generally won't cover an entire gene, but a fairly large region of the gene at least. And the other important thing is that all those 1,000 bases are completely contiguous. So they're all right next to each other. We're getting not data from just one spot within the gene, but all the information about that gene that we can. There are actually lots and lots of examples of clinical tests used with Sanger sequencing. It's really right now considered the gold standard for sequencing-based diagnostics. And that's primarily because of its low error rate. The ABI genetic analyzer from Life Technologies is really the dominant player here. There are probably one or two other minor platforms, but this is the one that people have been using and is generally considered the gold standard. So the first two technologies I talked about were very clearly genetic tests. Now we're moving to the range where it's pushing into genomics. Some people might not consider this a genomic test, but in general it would be. This is a focused array. And on the focused array, rather than looking at one particular locus or one particular gene, we're actually talking about looking at, say, maybe 1,000 or so different loci throughout the entire genome. And so you can see that as it's designated on the left. That means that we might look at one or even maybe a handful of bases within a particular gene. But again, the coverage is going to be very, very low, around 1,000 bases out of the 6 billion or so. These focused arrays are being used in the clinic. So we have the cytochrome P450 array from Roche using Affy's technology. This is for looking at drug response and dose response, helping to determine what the dosing should be for particular drugs, or whether certain drugs should be used or avoided. The other example that I've pulled up here is the Mammoprint, and that's from Agendia. And this is looking at breast cancer prognosis. And again, in both cases, we're talking about looking at a number of locations throughout the genome, but still using that single base multiplex technology of the focused array. Getting into what's much more comfortably called genomic technology or genomic diagnostic will be whole genome arrays. So again, like the focused array, we're looking at locations throughout the genome, but we're looking at a lot more. Instead of 1,000, we're talking about looking at 1 million or even up to 5 million. And so with that many markers, of course, they're going to be spread out across the genome. You're going to get multiple markers per gene. And so we see we're looking at markers within the coding regions, also in introns, not shown here, but you'd also be looking at intergenic regions, so regions that aren't connected with any particular gene. But again, the coverage is still pretty low, so the chances of having an immediate neighbor are quite low. Mostly you're just looking at one base per locus, and then you might just have multiple bases per gene. So the dominant platforms here, again, these are array-based. That primarily be from Illumina and Affymetrix. There are a couple others, but those are the primary players. And the main use for these arrays is really genome-wide association studies, or GWAS. The idea here is looking at those 1 million or 5 million or so SNPs or single nucleotide polymorphisms, differences between people, and trying to associate them with a particular phenotype, say susceptibility to diabetes 2 or something like type 2 diabetes. Um, uh, this has been used quite a lot. There's over 1,350 published studies so far. But these really primarily isn't directly used for diagnosis. This is primarily used as a research tool to discover the information that would be used to generate a diagnostic tool using a different technology. But GWAS has been extremely popular for a long time. This graph, which I realize is probably a little hard to read, is just showing the categories of all the different types of GWAS hits that have been found and how they're displayed or how they're distributed rather across the entire genome. So lots and lots of information there. Getting back to the sequencing technology, and specifically with next-gen sequencing, we have something called targeted sequencing. So here, similar to the whole genome arrays, we're looking at about 1 million bases or so, roughly. But in this case, rather than looking at 1 million discrete bases, we're looking at contiguous pieces. A very typical thing to do would be to focus on mostly the coding regions, but as these targeted sequencing products are usually customized, you can really target it to any particular region of interest. You could have these targeted regions spread out across the entire genome, or you could have them focused on a certain set of genes that are involved in a particular pathway. But regardless of how they're targeted, you're going to see that the calls are grouped together in these continuous bases. There are a number of ways that this targeted sequencing is being used in the clinic, generally for multi-gene tests. So the idea of rather than testing for one gene at a time, one condition at a time, to pool all this together into a single test that can test for many things. And that's really the power of moving from genetic testing to genomic testing. So in this particular example I'm showing you here, this is from Foundation Health. They have a test called Foundation 1, where they're looking at basically sequencing approximately 250 cancer-related genes. So if you have cancer, or if you have a patient with cancer, you can use this one test to sequence all the genes that are most likely to be involved in that cancer, either directly in causing it or genes that are affected by cancer. And so rather than trying to determine which particular gene that you might want to focus on, you can order this one test and get all the results across all the cancer-related genes right there together. Another example, another company is Invitae, and they're doing a similar kind of thing, but rather than focusing on cancer, they're actually trying to convert all of the single gene genetic tests that are available today into a single multi-gene sequencing-based test. So what they have right now is they have a test that covers, I think it's a little more than 200 genes covering about 260 genetic conditions. And what you can do is you can look at their catalog of all these conditions that they cover, and you can order one or two or a handful, or you can order all 262. But the price is actually the same. And right now that price is around $1,500. And the way it works is no matter what you order out of that catalog, they're going to run the same test. They're going to sequence across all those 200 genes covering all 262 conditions, and then just report back on whichever ones that you actually were interested in. And so it's up to the user, the physician, to decide whether they want to look at just a handful that are associated with what their patient needs, or whether they just want to run the whole test. And Invitae has plans to increase this. Like I said, right now it's about 262 conditions for $1,500. By 2014, they hope to have over 1,000 conditions and have the test cost under $1,000. So they're really trying to push hard on moving away from individual genetic tests to a single comprehensive genomic test, and then using informatics to help you decide whether or not you actually want to look at all of those or just a subset of them. So sort of moving up the scale in terms of complexity, we have exome sequencing, which really is a form of targeted sequencing. It's just the most extreme form that we have so far. and that's where we're targeting every single coding region of the genome. So here we're talking about 40 megabases, and as we've seen before with the other sequencing technologies, all these bases that are being sequenced are continuous. We're starting to get pretty significant coverage, so we're talking a little over 1% coverage of the genome. Still not full coverage by any means, but substantially more than any of the technologies that we've looked at so far. One step up from that, of course, would be the holy grail of a genomic test, and that's whole genome sequencing. And here I've just drawn a big red box over all the chromosomes because we're targeting everything. All the coding regions, all the intronic regions, all the intergenic regions, all the individual bases. Now, as a practical matter, the technologies that are available today can't really hit 100% of the sequence. They typically hit about 90%, and they cover that 90% pretty well. The other missing 10% is basically just harder. It's regions like the telomeres and the centromeres, or other regions that have a lot of repeat sequence that's just difficult to sequence through. And people are making improvements all the time, trying to up that 90% even higher and higher. But in general, that's what a next-gen sequencing test would cover. The question then is this massive technology? It's very expensive. It's still fairly time-consuming, and it certainly produces a lot of data. Is it able to be used in the clinic? And the answer is yes. It is already being used in the clinic. So the primary example that people are using this for is cancer patients. So cancer studies have used sequencing for a long time, but this is the idea of actually looking at the cancer cells of an individual patient, comparing cancer cells with normal cells, and trying to use that information to determine exactly what type of cancer it is. So it's kind of an old paradigm to say that people have either liver cancer or breast cancer or lung cancer. But that is not really the best way to look at it. What's really important is the molecular origin, the molecular nature of that cancer. What is the actual genomic lesion that's going on? And so this is an expensive procedure, so it's not usually the first line of defense. It's often used on patients who have stopped responding to other treatments, who have maybe an extremely rare form of cancer where there are no known treatments. But there are a number of cases where individuals have been sequenced and had their diagnostic treatment change, and actually have new drugs and new treatments applied to them that wouldn't have otherwise been even attempted because it wasn't known there was any evidence that it might work. But when you do this whole genome sequencing in cancer, you're actually getting a lot more information, and it's really helping the physicians push on and try to decide what to do with their patient. Now, this isn't a panacea. It's not easy. It's still a lot of data. It almost really should be considered more of an individual research project. It takes a lot of effort from a lot of people to really crunch through all that data quickly enough to help that patient. But people are starting to work on this, and they're starting to learn more and try to make this analysis happen faster so that it can become more routine. Another similar kind of use would be for rare childhood diseases. And again, these are cases where either there aren't any known treatments or treatments that have been tried aren't working. And so, again, almost as an individual research project, they will sequence the entire genome of that child and maybe even the genomes of the parents to do a trio analysis to really see what novel genomic lesions are present in that child that could help determine exactly what's causing the phenotype. And again, it runs the whole gamut of responses. There's been some where even after genomic sequencing, nothing has been determined, so it's unclear, all the way through being able to determine that relatively simple treatments can be applied. And in some cases, it's made just tremendous, dramatic improvements in the lives of these children. So again, it's still very expensive. It's still very complex and takes a lot of work, but it has been showing such dramatic results. And in this case, there's even we're even seeing reimbursement from the insurance companies because treating a child with these diseases and going through all the testing and on all the treatments can be extremely expensive. So even if the sequencing is $10,000 and say $20,000, $30,000 for the analysis, that can be much cheaper than other treatments or other testings, which can be in the multi-$100,000 range. So expensive as it is, it still can be considered um, feasible to do. The other area which has seen an explosion recently is the NIPT, or non-invasive prenatal testing. So in this case, rather than doing an amniocentesis, which is a fairly invasive procedure to directly look at the DNA from the cells of the fetus, in these tests, they actually take blood from the mother, and it turns out there's enough fetal DNA floating around in the maternal blood, and there's enough um, depth of sequencing available with these next-gen sequencing technologies, and there's enough sophisticated analysis that they can actually tease all that apart. And so as you can imagine, going from an invasive procedure to a non-invasive procedure is very attractive. So there's a number of companies that have been formed around this idea of doing NIPT testing, and it's been fairly successful and is gaining a lot of converts lately. So this is definitely an area of intense focus in the cross between next-gen sequencing and the diagnostic space. I see we have a couple questions coming in. I'll just remind everyone as they're listening to me, if you have any questions, please type them into that Q&A panel, and I will try to answer them when we're all done. So given what we've seen and this push towards genomic testing and maybe not away from genetic testing, but certainly as a complement to genetic testing, there are going to be some difficulties going forward. And one of those difficulties is certainly going to be analyzing the data. So this is just an overview of the types of data analysis that are involved in next-gen sequencing data. We have primary analysis, which is base calling and QC of the data. Secondary is assembling that data and aligning it. Tertiary is a group that would be calling the variants, annotating those variants, visualizing the data, and also maybe looking at some statistical analysis of the data. And then finally, we have reporting, and that's taking all that data and crunching it down to a single, hopefully easy to read report that can summarize everything that's been done. And laying over on top of all of that would just be the idea of data management, storing that data, moving the data around, understanding where it is and databasing it. So the primary data analysis has really been the purview of the vendors. And they've done a good job of this, and mostly people just kind of trust them with it and move on to the next one, which is secondary. Secondary analysis, or the assembly and alignment of DNA sequences, that was what the early adopters really focused on a lot. They spent a lot of energy. There's a lot of tools out there to do different, different algorithms for assembly and alignment, and trying to get a little bit faster, a little bit better. When you're talking about the mass market, they still cared about that secondary analysis, but they also started caring more about the tertiary analysis. Once you've aligned the data, I want to look at it. I want to see what variants are there. I want to look at the annotations of those variants, and I want to visualize it. But when we talk of going to the third major segment, the future segment of the clinicians and physicians, they're really going to care about the tertiary, and then even more so about the reporting. So they're still going to care about those annotations and make sure the annotations are good. But what they want is that final report. They want to hold it in their hand. They don't want to look at the terabase or terabyte of data that comes out of each sequencing project, but they want to just look at that report and say, here's my patient, here's what they have, here's what their genome looks like, and here's the information I need to help start making a diagnosis. And that's going to be a major push going forward. There are a number of new companies that have been formed that are really working very hard, both at making better annotations, but also making better presentations and better reports so that the physicians can get to that information as quickly as possible. So the question then becomes, what's the impact on patients? We've talked about the technology and who's using it, but who is it going to be used on? And there's a really big question right now about what exactly should the patients be allowed to see. should they have access to their entire genomic data, or should they only see the small bits that the physician has determined are relevant to the question at hand? And this is almost a religious debate. You see people arguing very strongly on both sides of this. Um, so it's by no means been resolved, but it is an active area of discussion. Of particular concern is this idea of the incidentalome. This would be the collected unexpected findings in a genomic test. So if you're doing a genetic test, this doesn't become an issue. If you're doing a simple test to determine how you're going to respond to a particular drug, you do that one particular test, you get that result. It's probably going to be a yes or no. That's all you find out, and you make a decision with your physician accordingly. You take a particular drug or you change the dosage of that drug. But when you start doing a genomic test, you can find out not just that one particular thing you were looking for, the handful of things you were looking for, you can find out everything. And you might find some things that either are just unknown. You might have some variants. You actually will have variants. You'll have several million variants. Many of them will have nothing associated with them clinically. We won't know anything about them. Some might be unknown, but in genes of known significance, and that might be disconcerting. You also might find some variants that are of known, that have known issues. For example, if you go in for a simple dosage test and you find out you have the defect in the Huntington's disease gene, that can be very unsettling. So it's unclear exactly what the best processes right now. Like I said, some people are arguing for full disclosure for everyone all the time. Others are arguing for a very protracted view of the information. So the ACMG has tried, if not to settle this argument, at least to bring some order to the discussion. And so they've kind of drawn a line in the sand and made a set of recommendations where they're saying that there are 56 genes for which any abnormal results should be reported to the patient regardless of the patient's wishes. So even without determining ahead of time from the patient whether they did or did not want to know these results, they're saying that these results should always be reported because they're significant enough and they're actionable, something that can be done with this information. So not everyone agrees with this, but it is nice to see this group come out with a set of recommendations, and it certainly serves as a point of discussion going forward. The other thing to ask, of course, is exactly how this genomic technology is going to impact single gene intellectual property. So I'm sure most of you have probably been aware of the Supreme Court case looking at Myriad's claims for BRCA1 and 2 testing for breast cancer. The results were interesting. There were 3 particular claims that were looked at, and basically what came about was a unanimous decision that for naturally occurring gene sequences, those could not be patented, and they weren't protected, so other companies or other individuals could use that information. This was generally seen as a big success, a big win for patients, and almost immediately a number of companies came out and either stated that they were immediately offering BRCA1 and 2 testing for Ambry Genetics and Gene by Gene were 2 of those. And then a handful of other companies said that they were, in the process of doing so. And so this, again, was considered a tremendous success, a tremendous win for patients, which is exactly what people wanted. But then almost as immediately, Myriad came back and said, well, no, that's not right. We lost that one particular case, but we have all sorts of other claims that weren't determined on, so we think we still have the rights to this. So it's certainly not a clear picture, but it's definitely difficult to imagine how single gene intellectual property is going to hold up in the face of genomic technology. So it's one thing when you're purposely trying to sequence a particular gene and that gene is protected, but it's equally difficult to understand how you could somehow have to block that gene information when you're sequencing your entire genome. So I'm sure we're going to see a lot more about this in the near future. So the other thing I want to just leave you with is what we've been talking about today is the genomic technologies, looking at next-gen sequencing for looking at the DNA. But it turns out, of course, our bodies are much more complex than that. We have the methylome to look at, so methylation and other regulatory sequences and structures to look at. We also have the transcriptome, looking at coding genes, looking at non-coding genes, and we even have the metagenome, so the collective genomes of all the microorganisms that are in your body. And these, of course, all of these are going to have medical impacts and are being looked at in the research realm, but will certainly be used in the diagnostic realm going forward. So we're just at the beginning of this, just at the very tip of the iceberg, if you will. So finally, I'll leave you with that. I just want to say that this, of course, is a very complex field, fast moving, lots of things happening all the time. So I would strongly encourage you to look at our website, BlueSEQ.com. We have a neutral repository of information. I will be answering questions here shortly, but if for some reason I don't get to your question or you think of one later, please feel free to reach out to me. I'm at sbaker@blueseq.com. And finally, for those who are interested in obtaining continuing education credits, there's a website that you can go to take the test to achieve those credits. Okay, with that, let's go ahead and see what kind of questions we have. So some people are asking about whether they can see this presentation later. My understanding is that all these presentations are going to be available as a recording for a full year after today. So you can certainly log back in and look at those. I'm also more than willing to share these slides with anyone who wants them. So if you actually want those slides, please just send me email to sbaker@blueseq.com, and I'd be happy to send them to you. Another question is talking about the idea of going from the sequencing cost today of roughly $10,000 to $1,000, and is that going to be a reality? I certainly think it will be. So, this $1,000 genome has been sort of something hanging there, kind of the prize that people have been trying to reach for a long time, and it's almost reached a mythical or magical proportion. But the truth is that $1,000 isn't really any more significant than the $4,000 that really a research genome costs today. So of course it's a little bit less money, but the point is the prices have come down so much that it really has transformed what people are doing in the biological sciences. So it's already been quite amazing. Of course, as it goes lower, that will just open up even new applications for people to look at. Personally, I think it will hit $1,000 genome, but I don't think it will stop there. There are certainly some of these far-reaching technologies which at least have the promise of going even lower to, say, maybe even $100 per genome. So the lower it goes, it's really just going to increase all the possibilities. Okay, so I think I've mostly got the questions answered there, but again, if you have any other questions you think of later, please don't hesitate to contact me. With that, thank you very much for your time, and I enjoyed presenting to you.